Bismillahirrahmanirrahim. My name is Muhammad Arif and I'm from class M380 2204C. And today I'm going to present about enhancing drought tolerance in transgenic tobacco through NAD1 defensing expressions. So what is that? So biotic and abiotic stresses negatively impact crop yield worldwide. Plant responds to the stresses by producing specific genes and secondary messengers to regulate their defense mechanism. Antimicrobial peptides or AMPs are crucial components of plant immunity, helping plants combat pathogens and maintain cellular health. AMPs can disrupt cell membranes, inhibit protein synthesis, and induce the production of reactive oxygen species. They are also involved in the plant responses to abiotic stress, such as drought and soil stress. NAD1 defensin is an example of an AMP that exhibits antifungal and insecticidal properties. Overexpressing NAD1 in tobacco plants enhance their drought tolerance. This finding highlight the role of AMPs in plant stress responses and their potential for improving crop resilience. So, for method and material, the coding of sequence of NAD1 was obtained from the NCBI database and a recombinant construct was designed and synthesized. The construct includes a signal peptide for protein secretion, a polyhistidine tag, and the CAMV35S promoter for overexpression in transgenic plants. The construct was cloned into a vector and confirmed through sequencing. The plasmid then digested and ligated into a expression vector. The resulting construct was transferred to Agrobacterium tumefaciens strain. Then the leaf explant of Nicotiana tabacum or the tobacco plants were inoculated with Agrobacterium culture and co-cultivated. The transformed leaf explant were then cultured on selective media containing the kanamycin and plant growth regulator for regeneration. The genomic DNA and RNA was extracted from transgenic and non-transgenic plants for molecular analysis. The presence of the NAD1 genes and its expression were confirmed using PCR and RT-PCR respectively. The production of NAD1 recombinant peptide was accessed through the protein extraction and SDS-based analysis. Next, for results. The recombinant expression vector was verified through digestion and ligation of the vectors. Transgenic plants carrying the NAD1 gene were characterized by PCR and RT-PCR. By confirming the presence of expression of the NAD1, the production of NAD1 recombinant peptide was detected in transgenic plants but not in the non-transgenic control plants. Photosynthetic pigment analysis show that transgenic lines exhibit a higher chlorophyll content compared to the control plants under the drought stress condition. Enzyme activity analysis reveal elevated levels of catalase and peroxidase and superoxide dismutase. Activities in the transgenic lines during the drought stress, the activity increased the transgenic plants under the non-irrigation state conditions. So, the findings suggest that the NAD1 defensin is involved in regulating drought stress response, and transformation with NAD1 can improve the plant tolerance to drought stress in tobacco plants. So, for discussion, the involvement of antimicrobial peptides AMP stress is not well understood, but certain groups of AMPs, including defensin, have been associated with the various abiotic stress. However, the role of defensin in drought stress is unclear. So, this finding suggests that NAD1 plays a role in maintaining photosynthetic pigments and activating an antioxidant system, thus improving the drought stress tolerance in transgenic plant. So, in conclusions, the overexpression of NAD1 antimicrobial peptide in transgenic tobacco plants enhance their tolerance to drought stress. The transgenic plants maintain a higher levels of photosynthetic pigments. Okay, that's all from me. Thank you.